Hello, and welcome to the JASN podcast, the monthly podcast where we highlight an exciting paper from the current issue of the journal. These papers may be clinical, basic, or translational, and today we have a basic but clinically translational paper that we're going to be discussing. I'm Dr. David Ellison, Professor of Medicine and Chemical Physiology and Biochemistry at Oregon Health and Science University, and a deputy editor of the journal. Today, I'm joined by Professor Ivana and Dr. Karla Márquez-Nogueras from the Department of Cell and Molecular Physiology and Cardiovascular Research Institute at Loyola University, Chicago. It's the Stritch School of Medicine, which is located in Maywood, Illinois, to discuss their paper entitled Cardiac Localized Polycystin-2 in the Natriuretic Peptide Sensing Pathway and Hypertension, which appears in the current issue of JASN. Doctors Kuo and Márquez-Nogueras, please introduce yourselves and tell us a little bit about your backgrounds.
Thank you very much for that invitation, Dr. Ellison. My name is Ivana Kuo. I am an assistant professor at Loyola, and I and my research team study the cardiovascular roles of polycystin and how they influence renal development and the cystic outcomes. My background is trained as a physiologist. I came from a background in actually neuroscience and then segued into studying calcium signaling pathways. And this is the part that I now focus on in my research lab.
Thank you, Dr. Ellison, and thank you to JASN for the invitation. My name is Karla Márquez-Nogueras. I'm actually a postdoctoral researcher in Dr. Kuo's lab. I currently work in understanding the role of polycystin-2 both in the cardiac setting as well as in the renal setting. I'm actually a scientist trained in microbiology. I was trained on the ortholog of polycystin-2 and understanding calcium signaling, and switch over now to the world of physiology.
Great. Well, welcome to you both. We're excited to have you here. So your paper described the possible involvement of the polycystin-2 gene in modulating the production and secretion of natriuretic peptides. While most of us are aware that autosomal dominant polycystic kidney disease is associated with excess cardiovascular morbidity, I had assumed that the latter was mostly the result of the chronic kidney disease that develops. How did you get interested in the possibility that there might be direct effects of the gene and the gene mutations on the heart? Was it primarily because of the data from the mouse models?
Yeah, so as you alluded to there in your question, there are cardiovascular morbidities associated with ADPKD. What was striking to me as I looked over the literature is that a lot of these cardiovascular manifestations appeared before the renal insult and the larger cysts forming within the kidney. And so this raised in our minds the question whether or not polycystins within the heart were actually contributing to the cardiovascular phenotype and in particular the hypertension that results. And so when I started out my lab about 6 years ago, I set out to answer that question. So using mouse models, we then were able to interrogate more carefully without the involvement of renal cysts, whether or not polycystins within the heart actually contribute to a cardiovascular manifestation.
Well, I think the first major finding that I noted in the paper was that both the systolic and diastolic pressures were elevated in mice with polycystin knocked out only in the heart. And the difference appeared to me to be biologically significant. Was this consistent with your pre-existing hypothesis, or was this an unexpected result?
Yeah, this is the joy of science. This is one of the fun parts about setting up a hypothesis and being open to where the data takes you. So originally, when we set out to investigate what polycystins were doing in the heart, we had set up the hypothesis that it would result in heart failure. And actually, in our early studies, we kind of didn't see that happening right off the bat. But to kind of complete the study, we then interrogated what was going on with the blood pressure. And lo and behold, this is where we saw this very profound, as you note, rise in both the systolic and diastolic pressures. Now, the association with natriuretic peptides wasn't completely a path that we didn't think about, because in prior studies using polycystin-2 heterozygous mice, which are whole body heterozygous, we had previously noted, or at least in my postdoctoral work, that those mice did not have the same amount of natriuretic peptide response. We didn't measure the blood pressure, but we kind of had a hint from those studies that perhaps this could be the pathway that we should go down and delve further. So overall, to say that it was an unexpected bonus, dare I say, that these mice were hypertensive.
I couldn't agree more that that this is one of the joys of science, is coming across an unexpected observation and then pursuing it and really learning something new. So the NPPB transcript levels, which I guess reflect transcription of natriuretic peptides, were somewhat lower in the knockout mice than in controls. And yet the circulating natriuretic peptide levels were similar in the two groups. As circulating natriuretic peptides would be expected to interact with receptors in the kidney and the vasculature, does it seem surprising that there was no difference in the circulating levels of natriuretic peptides?
Yeah, that's a good question. And yeah, we were a little bit surprised that we did not see circulating levels change. We were expecting, because the mice were hypertensive, and we know that when we have hypertension, natriuretic peptides increase, and that's not what we found. We know that when we looked at the transcription levels, we already saw that there was impairment in that pathway, but we didn't see that being translated into a decrease in natriuretic peptide production. So the possibility as to why we're not seeing changes in that is because we know that other tissues, like the brain, can still produce natriuretic peptides. So what we're hypothesizing is that there is a possibility of other non-cardiac sources still producing natriuretic peptides. And that's why in the polycystin-2 knockout mice, we're not seeing those levels increase or change, basically.
Well, you mentioned something that I may have heard before, but I had forgotten, that the adrenergic system, adrenergic stress, activates natriuretic peptide production, possibly through chromogranin B-related mechanisms, and also involving NF-kappa B. Could you explain more about how you imagine the polycystin-2 is involved in this signaling pathway and how knocking it out disrupts it?
Yeah, so NF-kappa B is known to associate with I-kappa B, and the association of these two molecules is what sequesters NF-kappa B into the cytosol. We know that upon calcium increase in the cytosol, these two proteins dissociate because it phosphorylates the I-kappa B, and that causes it to be degraded. That disassociation allows for NF-kappa B to be translocated to the nucleus, where it can then regulate gene transcription, like NPPA and NPPB, which produces AMP and BNP. In our model, what we hypothesize is that PC2 and the calcium influx that is regulating via the IP3 receptor, as well as producing local calcium signals, because polycystin-2 is a calcium channel in itself, is allowing for this calcium to regulate these molecules, which then allows for the transcriptional regulation. So when you knock out polycystin-2, we are missing the regulation of both the calcium release from the IP3 receptor and its local calcium signals.
Well, I think one of the parts of the paper I found interesting was that you also used a cell model to examine some of these issues, and you found that deletion of polycystin-2 in C2, C12 myoblast cell model reduced NPPA expression, and that the expression was restored then when you reintroduced polycystin back into the cells. And from my reading, this seemed to provide further support for a role of polycystin-2 in regulating natriuretic peptide production. But it wasn't entirely clear to me whether patients with mutations in polycystin-1 might also have deficiencies in natriuretic peptide secretion. They certainly, as far as I know, also have an excess incidence of hypertension and cardiovascular morbidity. Could you comment on that question?
You're entirely correct in that. And in fact, the manifestations in terms of the disease are more pronounced in PKD1 patients than PKD2 patients. And we also know that PKD1 associates with PKD2, or polycystin-1 associates with polycystin-2. In terms of animal models, this has not been directly investigated. However, what I can say is that Joe Hill's group has looked in the polycystin-1 cardiac-specific knockout mouse model from birth and has incidentally reported that natriuretic peptides are down in their model, but didn't necessarily follow up with subsequent experiments, for example, with blood pressure, or to see if mechanisms within the signaling pathway of natriuretic peptides are affected. So I would say watch this space, because this is an area of active investigation in our laboratory. And I think getting back to the clinical implications, given that so many patients with PKD have those PKD1 mutations, and given the association between PKD1 and PKD2, it seems very logical to make that connection that PKD1 patients do indeed have a similar pathway that is being affected in terms of the natriuretic peptides.
And in the heart, do you envision that it's a PKD1, PKD2 heteromer that's present? Is that what you think is going on?
It might be a little bit more complicated than that. I think from the cryo-EM models, there is certainly evidence that polycystin-1 and polycystin-2 can associate to form channels. What we have seen is that, at least by careful examination of the location of polycystin-1 and polycystin-2, they perhaps can locate in different areas of the cardiomyocyte. So one of the things I would comment about the kidney field is that polycystin-1 and 2 are often found in association with the primary cilia in renal epithelial cells. Cardiomyocytes, if they're not on the endocardial surface, don't have primary cilia. So it then raises the question, where are these molecules located? And at least in our hands, we seem to see that polycystin-2, at least in cardiomyocytes, has an association towards perhaps some of the sarcoplasmic reticulum as well as the ER. And whereas polycystin-1, I would kind of say, watch this space. So giving that a little bit of a teaser. But I think what you're pointing at is that there is very strong hints of an association there.
Well, I know you're both basic scientists, but let me pull you back a little bit into speculating about clinical relevance. So I'm a clinician in addition to being a scientist, and the most exciting aspect of the study for me was really the therapeutic implications. And so I wondered if low production of natriuretic peptides contributes to early hypertension in PKD patients, maybe PKD1 and 2, perhaps. The use of the ARNI agents that both increase natriuretic peptides and block angiotensin receptors has appeal because it might address several problems. Certainly in PKD, we know that treatment with drugs that interfere with the RAS system is beneficial, but maybe adding a component of activating the natriuretic peptide system would be helpful. Can you talk about that a little bit and what you think about therapeutic implications?
Yeah, so definitely that we're hoping that this basic research provokes discussion amongst the nephrologists. So as you allude to, combination treatment, that combined neprilysin inhibitor, neprilysin is a circulating molecule that breaks down natriuretic peptides. The neprilysin inhibitors combined with ACE inhibitors, ARBs, are clinically used for heart failure patients. As far as I'm aware in the literature, and also talking with other nephrologists, utilization of neprilysin inhibitors has not been applied to PKD patients. So we're hoping that, that our studies does provoke a discussion amongst the nephrologists. I would also comment that the HALT PKD trial, which tested the efficacy of fairly aggressive ACE inhibitors combined with ARBs, did reveal that there was a small group of patients that were on the high dose of ACE inhibitors and ARBs that actually kind of reached the ceiling in terms of their blood pressure effects. So I think that there might be that little window of opportunity there for these neprilysin inhibitors to be used. I should also comment that in our mouse model, we are knocking out both alleles of polycystin-2.
Yeah.
Whereas in patients, right, patients are born heterozygous. In other words, they have a germline mutation, and then whether it's a somatic hit or something else that brings down the level of the other copy in the kidney, then results in cysts. So what we're talking about in patients truly is actually going to be the situation where the cardiomyocytes don't probably have a complete deficiency of the natriuretic peptides, but they probably have a muted response in the production of natriuretic peptides, and thus even bolstering something that is lowly produced with neprilysin inhibitors could be a way to go forwards.
Yeah, well, it sounds like you'll need to look for some collaborations from the clinical side to explore some of those things, but I think that sounds like a very exciting path to go in the future. So really appreciate your sharing some of the results of this exciting work. I think it's beautiful work, and you should both be congratulated. And I look forward to, as you say, there's more to be coming about this space, and I look forward to reading more, perhaps in JASN. So thank you both very much.
Thank you very much.
Thank you. Thank you very much.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.